Precipitation tests. Precipitation reactions are based on the interaction of antibodies and soluble antigens. The two soluble reactants interact to make one insoluble product, the precipitate. Precipitate settles to the bottom of the tube in case of aqueous solution or appears as an opaque white line in case of gel. Antibody that aggregates the soluble antigens are called precipitants. Precipitation tests are sensitive in detecting very minute quantity of unknown antigens, but are relatively less sensitive for the detection of antibodies. The amount of precipitate formed is greatly influenced by the relative proportion of antigens and antibody and avidity of an antibody. Precipitation reactions can be broadly of three types, that is precipitation in solution, precipitation in agar, and precipitation in agar with an electric field. The zonal phenomena. In quantitative precipitation test, normally the quantity of precipitate formed should be in a decreasing trend with increasing dilution of antibodies. However, at times, no precipitation is observed at higher concentration of antibodies followed by appearance at higher dilutions, reaching to the peak and then again decreasing with increasing dilutions. This is referred to as zonal phenomena, in which precipitation does not take place in zones having excess of either antigen or antibody. Maximum precipitation occurs when both the reactants are in optimal proportions, that is, at a zone of equivalence. Initial zone with no immune complexes is known as pre-zone or prozone. Middle zone with highest concentration of immune complexes is called zone of equivalence, and last zone with decreasing concentration of immune complexes is called the post-zone. The prozone phenomena. One of the important features of precipitation reaction, as we have already seen, is that the reaction occurs optimally at zone of equivalence. On either side of this, precipitation is actually prevented because of an excess of either antigen or antibody. Prozone phenomena is a false negative response resulting from higher antibody titer. Excess antibodies interferes with the formation of antigen-antibody lattice necessary to visualize a positive precipitation test. Merak in 1934 proposed a lattice hypothesis to explain the prozone phenomena. The hypothesis is based on the assumptions that each antibody molecule must have at least two binding sites and an antigen must be multivalent. Precipitation tests include ring precipitation test, slide precipitation test, gel diffusion precipitation, immunoelectrophoresis, countercurrent immunoelectrophoresis, and rocket electrophoresis. First is ring precipitation test, is also known as tube flocculation test. In this test, the antigen solution is layered over antiserum in a small tube or a capillary tube. Precipitation between antigen and antibody in the solution is marked by the appearance of the ring of precipitation at the junction of two liquid layers. It is used for grouping streptococci on the basis of C polysaccharide by Lancefield method, and for determining C-reactive proteins, and in VDRL, that is Viral Disease Research Laboratory test. Slide precipitation, also known as slide flocculation test. In this method, take a drop of antiserum onto the clean glass slide. Over the antiserum, a drop of test antigen is added. This is followed by thorough mixing. If clumping between the antigen and antibody occurs, the slide test will give a positive result, and vice versa. Floccules can either be seen by naked eyes or demonstrated well under the microscope. The example in which such test is followed is VDRL test for syphilis and Kahn's test for syphilis in tubes. Gel diffusion precipitation, also known as immunodiffusion. The precipitation carried out in gel is known as immunodiffusion. Reactants diffuse out and form an opaque white line of precipitate where they meet in optimal proportions. Gels are prepared from 1 to 2% agar or agarose solution in a suitable buffer such as barbiton buffer. Speed of movement can be increased by applying the electric current. Rate of diffusion is influenced or affected by the factors like the particle size of the reactants, temperature, gel viscosity, interaction between gel matrix and reactants. The preferred concentration of agar is between 0.3 to 1.5% of the effective dilution of the reactant to form a lattice. In agar, diffusion takes place by either of the two ways, single diffusion or double diffusion. Single diffusion precipitation. When antigen diffuses in the agar medium, antibodies homogeneously already spread in the agar. It is carried out either in the tube, single gel diffusion by Odin, or in a slide, that is single radial immunodiffusion by Mancini. The principle of the reaction is the antigen is placed in the well cut in an agar gel containing suitable diluted antibody. A ring of precipitate forms where the reactants meet in optimal proportions. The higher is the concentration of the examined antigen, the greater is the diameter of the ring. This type of immunodiffusion is used for quantitative determination of immunoglobulins, complement components, and other serum proteins. Odin immunodiffusion method. It is a type of single immunodiffusion method which occurs in one dimension. In Odin immunodiffusion, antibodies are added in the molten agar media and poured onto the petri dish or tube. After solidification of the gel matrix, a layer of soluble antigen is added. As a result, the incorporated antibodies in the gel matrix will not diffuse, but the diffusion of antigen will occur. The antigen will diffuse towards the antibodies and form the line of precipitate. Radial immunoelectrophoresis, where the antibodies in the serum are added in the molten agar media and poured onto the glass slide. After the solidification of gel matrix, create wells and add test antigen into it. Upon incubation, the antibodies incorporated within the gel will react with the specific antigens. A radial diffusion occurs, that is, the ring of precipitation forms exterior to the well surface. The diameter of the precipitant ring is directly proportional to the concentration of antigen. Radial immunoelectrophoresis is a rapid and more sensitive method for quantitative measurement of antigen. It is a type of single diffusion method which occurs in two directions. Interpretation of radial immunoelectrophoresis. The diameter of the ring is proportional to the concentration of antigen. Generate standard curve using the known concentration of antigen and add unknown sample. Read from standard curve. Double diffusion. Double diffusion consists of the diffusion of both antigen and antibody on the solid agar media. It occurs in two ways, double diffusion in one dimension and double diffusion in two dimensions. Double diffusion in one dimension is also known as Ouchterlony immunodiffusion method, and double immunodiffusion in two dimensions is also known as Ouchterlony technique. Ouchterlony immunodiffusion method. It is a double diffusion in one dimension. Example in case of Ascoli's precipitation test. In this method, the antibody is incorporated in the agar gel in a test tube above which the layer of plain agar is placed. The antigen is then layered on the top of this plain agar. During incubation, the antigen and antibody move towards each other through intervening layer of plain agar. In this zone of plain agar, both antigen and antibody interact to form the band of precipitate at their optimum concentration. Double diffusion, double immunodiffusion by Ouchterlony. It is a type of double immunodiffusion in two dimensions. Antigen and antibody are allowed to diffuse towards each other in an agar medium. When antigen and antibody meet in optimal proportion, they produce a thin line of precipitate. Position of the precipitate line depends on concentration of both antigen and antibody and on the diffusion coefficient. This reaction is used for diagnosing various bacterial, viral, fungal, and autoimmune diseases, and also for recognizing toxins production by Corynebacterium, diphtheria. The technique includes the test is performed by cutting wells in the agar gel poured on the glass slide or in a petri dish. The antiserum consisting of an antibody is placed in the central well and different antigens are added to the well surrounding the center well or vice versa. After an incubation period of 12 to 48 hours in a moist chamber, the lines of precipitin are formed at the zone of optimal concentration of antigen and antibody. Precipitation in agar with an electric field includes three methods, that is immunoelectrophoresis, countercurrent immunoelectrophoresis, and rocket immunoelectrophoresis. Immunoelectrophoresis. The term was first coined by Kreppler and Williams in 1953. It is a process of combination of immunodiffusion and electrophoresis. It is a powerful analytical technique with high resolving power as it combines the separation of antigens by electrophoresis with immunodiffusion against an antiserum. In this method, first the agar is overlaid on a glass slide. A mixture of antigen is added into the well cut into the agar. During electrophoresis, antigen moves in the electric field according to their charge and size. A trough is then cut into the agar gel parallel to the direction of electric field. Antiserum containing a mixture of antibodies is then added to the trough. Antibodies for all antigens in the mixture diffuse at a right angle to the previous direction of electrophoresed antigen. The presence of elliptical precipitin arcs representing reaction between individual proteins with its antibody. The absence of the formation of precipitate suggests no reaction. Different antigens, that is protein, can be identified based on the intensity, shape, and position of the precipitation lines. Application of immunoelectrophoresis. The test helps in the identification and approximate quantization of various proteins present in the serum. It is used to analyze complex protein mixtures containing different antigens. Immunoelectrophoresis is used in patients with suspected monoclonal and polyclonal gammopathies. It is also used to detect normal as well as abnormal proteins such as myeloma proteins in human serum. It is an older method for quantitative analysis of M proteins in serum and urine. The advantages of immunoelectrophoresis is that the number of antigen can be identified in a sample. Limitations of immunoelectrophoresis is that it is slower and less sensitive. Also fails to detect some small monoclonal M protein because the most rapidly migrating immunoglobulins present in the highest concentration may obscure the presence of small M proteins. Countercurrent immunoelectrophoresis. It is a rapid and more sensitive variant of double diffusion method in 2 dimensions. This method was used to detect hepatitis B surface antigen. It is used for the rapid detection of bacterial antigens in clinical samples. The agar gel is poured over the glass slide, create wells on both the edges of the glass slide. Antigen is placed in the cathodic well, that is negative N, and antiserum is placed in the anodic well, that is positive N. Both antigen and antibody are made to migrate towards each other by electrophoresis. The antigen will migrate towards the anode and the antibody will migrate towards the cathode. On the interaction between the antigen and antibody, the precipitin line will be formed. Rocket immunoelectrophoresis. This technique is the modification of radial immunodiffusion using electrophoresis developed by Laurel. It is called so due to appearance of a precipitin bands in the shape of cone-like structure, that is of rocket appearance, at the end of the reaction. Antibody is incorporated in the gel and antigen is placed in wells cut in the gel. Upon electrophoresis, antigen being negatively charged will migrate towards the positively charged antibodies. This results in the formation of precipitin line which has the shape of rockets. The height of the rocket measured from the well to the apex is directly proportional to the amount of antigen in the sample. Rocket electrophoresis is mainly used for the quantitative estimation of the antigen in serum. Thank you for watching. Don't forget to subscribe and hit the notification bell to remain updated with more such videos.